Dear laureates, ladies and gentlemen, boys and girls, each revolution has its icons. The Industrial Revolution is inextricably linked with the steam engine, the French Revolution with the guillotine. The scientific revolution, I think, is best symbolized by the lens, because the lens opened new worlds to us. It made the invisible visible. It showed us things that had never been seen before, and more importantly, it showed that there are things invisible to us that may be accessed if we just had the ingenuity to find out how. In Italy, Galileo Galilei turned his lenses upwards towards the very big, the very large. He looked at heaven and he was told to go to hell. In the Netherlands, Antoine van Leeuwenhoek took his lenses and pointed them downwards towards the very small. Now, there was no current understanding of the microscopic world, and so he avoided the ban. But in doing these observations, the science of observational astronomy was changed forever. Microbiology appeared as a new subject, and science was changed into what we know it of today. In 1874, the inorganic chemist Christian Wilhelm Blomstrand was observed walking on the university grounds in Lund, a small town in southern Sweden, bitterly crying because he had read a paper published the year before by AB showing that the ultimate resolution of a light microscope was on the order of the wavelength of visible light. And Blomstrand realized that he would never be able to see an atom. Now, strangely enough, the ability to see single atoms appeared long before anyone was able to circumvent Abbe's diffraction limit. Electron microscopes, very powerful machines, can look at objects so small that it defies our understanding. But they are of little use if you want to study living systems. For living systems, you need visible light. And what this year's laureates have accomplished is to take the light microscope that uses ordinary light and convert it into a machine that can peer so far deeper into matter than before. This year's laureates, Stefan Hell, William Moerner, and Eric Betzig, will tell you about their journey into the nanoscopic world. And first out and onto stage is Stefan Hell, born in 1862 in Arad, Romania, taking his PhD at 1990 from the University of Heidelberg, and now a director at the Max Planck Institute for Biophysical Chemistry in Göttingen, and division head at the German Cancer Research Center in Heidelberg. And it must be very fitting that the man who begins these lectures reminds us of the fate of Galileo, but his name really means light. Welcome onto the stage.
Thank you very much, Sven. Ladies and gentlemen, I think we're all familiar with the notion a picture is worth 1,000 words, a seeing is believing. And this not only applies to our daily lives, it definitely applies also to the natural sciences, as we just heard. Because I don't think it's a coincidence that the beginning of the natural science in history very much coincides with the invention of the light microscope. Because with the light microscope, we were able to see for the first time that every living being consists of cells as basic units of structure and function. And with the light microscope, bacteria were discovered and mitochondria as subcellular organelles. However, We learn at school that the resolution of a light microscope is fundamentally limited to about half the wavelengths of light, which typically amounts to about 200 nanometers. And if you want to see smaller things than that, we have, such as viruses, for example, we have to resort to electron microscopy. Now, electron microscopy achieved a much higher spatial resolution, 10-fold, 100-fold, 1,000-fold, in fact, down to the size of a single molecule. And therefore, the question comes up, why do we care for the light microscope and its spatial resolution now that we have the electron microscope? Now, the answer to that question is given in the next slide, where I made a little experiment. I counted actually the numbers of papers in this journal where a light microscope was used and where an electron microscope was used. And now you see which of the two systems won. It's still the case today that light microscopy is the most popular microscopy technique in the life sciences. And that is for two strong reasons. The first reason is that light microscopy is the only way by which you can look inside a living cell in 3 dimensions, or even living tissue. So it's minimally invasive. But there's another reason. If you want to look in a cell, usually we are interested in certain proteins or other biomolecules, and we have to make them distinct from the rest. We have to highlight them, because to light or to electrons, all the proteins look the same. And in light microscopy, this is easily possible by attaching a fluorescent molecule to the protein of interest, for example. And that fluorescent molecule has a very interesting feature. It has 2 states, an energy ground state and an energy excited state. And if you shine light on it, for example, green light, it can absorb a green photon, and then what will happen, the molecule will be raised to a higher lying state, then its atoms will wiggle a bit, that's why we have these vibrational substates. Then after about 2-3 nanoseconds or so, the molecule relaxes by emitting a fluorescent photon. Now, because some of the energy of the absorbed photon is lost here in the wiggling of the atoms, the fluorescent photon is redshifted in wavelengths, it's orange in here. But that's good, because then we can easily separate it from the excitation light, so from the light with which the cell is illuminated, and that makes fluorescence microscopy extremely sensitive. In fact, it can be so sensitive that you can detect a single molecule, as has been discovered by our co-laureate W. Moerner. However, if a second molecule, a third molecule, a fourth molecule, a fifth molecule comes closer together than to the first, than 200 nanometers, we cannot tell them apart, because they appear in the microscope as a single blur. So resolution is about telling things apart, telling small features apart and distinguish them and detect them as separate entities. And now you can imagine that because fluorescence microscopy is limited to 200 nanometers, a lot of information is lost if you look with a fluorescence microscope into a cell. So anything that is below the scale of 200 nanometers appears blurred. So therefore, if we manage to come up with a light microscope or a fluorescence microscope that has a much higher spatial resolution, that would have a tremendous impact in the life sciences in particular. Now we have to understand first why the resolution of a light microscope is usually limited. In simple terms, it can be explained as follows. The most important element of a light microscope is the objective lens. And the role of this objective lens is nothing but to concentrate the light in space, to focus the light down to a point. However, because light propagates as a wave, it's not possible for the lens to concentrate the light on a single point. Rather, the light will be smeared out here in the focal region, forming a blob of light that is at least about 200 nanometers wide and about 500 nanometers along the optic axis. And this has a major consequence. If we're having several features falling within this range, of course, they will all be flooded at the same time with this light, with this excitation light. And hence, they will produce signal at the same time. And if you try to detect this, the signal with a lens, and put it onto a detector, the signal produced by these features within this 200 nanometer blob will be confused. So if you look at this one, it produces this blob, and the other one that blob, and so on. And so no detector will be able to tell the signal here apart, no matter if it's a photomultiplier, the eye, or even a camera. Now, the person, as we heard, who discovered that diffraction poses a serious limit on the resolution was this man, Ernst Abbe, who lived at the end of the 19th century, and he coined this barrier in an equation that is named after him. It's saying that in order to be separable, two features of the same kind have to be further away than the wavelengths divided by twice the numerical aperture of the objective lens. And you can find this textbook basically, this equation basically in any textbook of physics, of optics, but also in textbooks of biochemistry and molecular biology because of the enormous relevance of light microscopy to this field. But you will also find it on a memorial that was erected in Jena, in Germany, where Ernst Abbe lived and worked, and there it is written in stone. And this is what scientists believed throughout the 20th century. Not only they believed it, it also was a fact. For example, if you want to look at cytoskeletal features in the 20th century, this was the type of resolution that you got. But now, today, we get this, and this is a standard. And so what I'm going to talk about is, I would like to show you how this transition was made actually from the standard previous resolution to this type of resolution. Now it started out in the late 80s. I was a student in Heidelberg in those days. And I did light microscopy, so I of course was familiar with this equation. And I said to myself, how does it come? This equation was coined in 1873, but now we have 1990. So much new physics actually evolved in the 20th century. So many new phenomena were discovered. I had to learn so much for my examination. There should be some phenomenon, at least one, that could be used to overcome this diffraction barrier. Well, I understood that it won't be just by changing the way the light is propagating, the way the light is focused. It must have something to do with the major discoveries in the 20th century. Quantum mechanics, molecules, molecular states, and so on. And for that reason, I started to check my textbooks again in order to find something that could be used to overcome the diffraction barrier in a light focusing microscope. And one day I put it down in writing. So the idea was, check out the properties of flow of force, state properties, and so on. Or maybe there's a quantum optical effect that could be used to overcome this barrier. And so one day, when I was not very far from here in Orbu, in Turku, just across the Gulf of Bothnia, on a Saturday morning, I opened this textbook and stumbled across this page, and that this was a page about stimulated emission. And all of a sudden, I was electrified. And now I explain to you why I was so electrified. The problem, as I said, the lens concentrates the light in space, but not better than 200 nanometers. And all the features are flooded simultaneously with excitation light. We can't change that. Sure, we can't change that. But maybe we can change the fact that all the features that are flooded here with excitation light are in the end capable of sending light to the detector. Because if we manage to keep some of them dark, the dark state to be precise, such they are not able to send light to the detector, we can separate the bright state molecules from the dark state molecules. In this case, we can tell this one from that one apart, or that's the top view. So the idea was to keep molecules in a dark state. You see, the state is the essence. Now you will see, or say, are there dark states in a dye? Well, let's have a look at an energy diagram. There is the ground state, as I explained, that's the excited state. And for the molecule to emit fluorescent light, it has to be in the excited state. So this is the bright state, but the ground state is, of course, the dark state. Because if the molecule is in the ground state, it won't emit light. So there is a dark state, that's the ground state. Now guess what stimulated emission does? It does nothing but making ground state molecules, dark state molecules. And that was the reason why I was so excited. I found a way to make dark state molecules just with normal flow of force. And now you can imagine how the microscope works, STED microscopy. Well, this is the lens, of course, and we have a sample and a detector. And then, of course, we use a beam of light for getting molecules from the ground state to the excited state, so making them bright. And of course, this light will be diffracted here. We'll get this blob of light of about 200 nanometers. And all the signal that is produced here will be going actually to the detector from all the molecules. But now, we use a beam of light that induces the stimulated emission, makes dark state molecules. And the idea is now, of course, that to push the molecules, in the case they get excited, instantly back down to the ground state so that the molecule is not capable of emitting light because it has to assume the ground state instantaneously. Now, the physical condition for doing that is that the wavelengths here of the stimulating beam is redshifted. It has a lower photon energy so that we don't excite molecules, but couple the molecules from this state back to this ground state. That's one condition, but that's not the only condition. There's another condition. Of course, we have to make sure that there is a red photon at the molecule that really pushes the molecule down. Most photons pass by because there is a finite interaction probability of the photon with the molecule. But if you have a certain threshold intensity, you can be sure that it's at least one photon that kicks the molecule down and makes it instantly dark in the case it gets excited. So what you see here is the intensity of the red beam, and this is the probability of the molecule to still assume the excited state, the S1. And now you see after a certain threshold intensity, you've turned it off. You can put as green light as much as you want, but it won't be able to emit. And now the game is easy. We just convert this beam such that it doesn't form a pattern in here, but a ring. And then we can turn off these molecules, as you can see, and here, of course, the ring or donut will become weaker and weaker, and here it is ideally 0. So here we won't be able to turn them off. But let's assume now that we want to see just this fiber. So we have to turn off this fiber and that fiber. What do? Well, we can't make the ring smaller. That's also limited by diffraction. I would say that's not possible. But we don't have to do that. We just have to shut off molecules because this game is about states. And so what do? We make the beam strong enough so that the molecules here are essentially turned off because they're confined to the ground state. Because even now, even here, in absolute terms, the intensity is beyond the threshold. Now we're there. Only here the molecules are allowed to emit, and we can separate the signal from the neighboring fibrous signal. And now we can take pictures, sure. We can move the beams across the specimen, and now we can separate this fiber from that fiber and that fiber, because the molecules are forced to emit sequentially in time, because we play this on-off game. And then, of course, we move on and so on. I'm just highlighting it again so that you understand it very clearly. This is the excitation region, but only this subclass of molecules are allowed to emit. Those here are kept in the ground state, effectively. Whenever you make a check which state they are, you will always find them in the ground state. And we move on, and then, yeah, I'm putting back the donut so that it's clear that I'm using the donut, of course. So, and this turned out to work. Now, this is a standard recording of something that you cannot really see what it is. But now, I'm using STED microscopy, and I see that because the resolution is higher, roughly by an order of magnitude, we can clearly discern actually what's imaged in here. So these are nuclear pore complexes, and as a result of the high resolution, it's about 10-fold better than previously. You can see that this nuclear pore complex consists of about 8 molecules as molecular subunits, and you have this 8-fold symmetry coming out very clearly. Comparison with the standard resolution looks like this, and then of course, that's almost no comparison. So, if you have this increase in spatial resolution, of course you get new information. There's no doubt about that. And I would like to show you an application where something new was learned with this microscope. So this is an application which was done in collaboration with virologists, with people interested in HIV. Now, viruses are about 30 to 150 nanometers in diameter usually. And of course, you want to image them with a light microscope. No way, you won't see any protein distribution on them. It'll just be a blur. Now, HIV is about 140, and in this study, the scientists were interested in finding out how this protein called ENF is distributed on this HIV particle. Normal recording, you cannot see anything. High-resolution recording STED, in this case, you see that it forms patterns on the HIV, and what has been found out in this study is that the mature HIV particles, so those that go to the next cell, infect the next cell, have their ends in a single place. So that's a requirement in order for HIV to be very effective. So that's a new insight that was gained as a result of high-resolution imaging. Of course, a strength of light microscopy is that you can image living cells. And this is an example of video rate recording STED microscopy. These are synaptic vesicles in an axon of a living neuron. You can see how they move about and learn about their dynamics and what were their fate in time. And that's important to be able to image living cells. But a live cell imaging at the extreme, so to speak, is this one. Here we open the skull of a mouse. It's an anesthetized mouse, and look into the brain of the mouse at the molecular layer, the visual cortex. Now, this is a transgenic mouse, so some of its neurons expressed a fluorescent protein, and that's why we can see this neuron quite clearly in here. The rest of the brain tissue is dark. And now we can take sequential recordings and see that actually those are the receiving ends of the neurons. These are the so-called dendritic spines, and they also slightly move. It's worthwhile zooming in. As you can see in here, this is the spine neck, and this is cup-shaped receiving end. And the message here is that this sub-diffraction resolution imaging, STED, for example, allows you to see these tiny morphological changes. And I'm very confident that in the not-too-distant future, we will be able to image the proteins here at the synapse and perhaps even give a visual cue on the mouse and see how this changes actually the protein distribution directly on the synapse, and in the end learn how neuronal communication works and perhaps memory formation. Now, this is just another example. Of course, one can take three-dimensional imaging because you can focus into the brain tissue, for example, and this is a 3D super-resolution recording of actin in a living neuron in a so-called organotypical hippocampal slice. Now, with that, I'm coming back again to the basics, to the resolution. Some of you will ask, what is the resolution that you can get? What's the limit? Is there a new limit? So let's get back to the principle. Well, the name of the game is that we turn off molecules in here. And if we make the region smaller in which the molecules are still allowed to emit, of course, the resolution gets larger. That's clear. So it cannot be described by this equation anymore, very clear. In fact, it must depend on the intensity that is found here on the donut crest and on the threshold intensity, which is a characteristic of the photon-molecule interaction. And so the larger this ratio gets, of course, the smaller the d will get. And so you can imagine that it must be found here in the denominator if you describe it with obviously new equation that is required now. In fact, d scales inversely with the square root of I over Is. So the larger this gets, the smaller d will get. And in principle, of course, in this equation, it goes down to 0. Because if this becomes very large, d goes down to 0. Now here you can't separate the two molecules because both are allowed to emit at the same time, but let's make it a bit stronger, the beam, so that only one molecule fits in. And now you see the resolution limit, so to speak. It's the size of a molecule. Because this molecule is, of course, the smallest entity that you can separate. This is not surprising. We separate by having molecules in two different states. And so it must be the molecule, which is, in the end, the limit of the spatial resolution. So in this cartoon, I'm having two molecules coming very close together. And now we can separate them, because at the time this one is emitting, the other one is off. By the way, if this is totally dark, one photon is enough to know that there is a molecule, because we know the position of the molecule by the presence of the red photons. They determine exactly where the molecule is allowed to emit and where it's not allowed to emit. Now you will ask, do you get molecular spatial resolution typically, for example, here and there in a cell? Well, with STED microscopy right now, the standard of resolution is between 20 to 40 nanometers, depending on the fluorophore, depending on the chemical environment, but that's something that is progresses. And I would like to show you that, of course, if you have the right fluorophore that can be turned on and off as many times as you want, you can do better. In fact, there are fluorophores, not organic ones, inorganic ones, that do that already. So these are so-called charged nitrogen vacancies in diamonds. So these are fluorescent defects found in solids in diamonds, and they can be turned on and off almost as many times as you want. And here we managed to squeeze down the region of emission to 2.4 nanometers. Keep in mind, the wavelengths responsible for this is 775. So it's smaller than 1%. So it's a small fraction of the wavelengths. Now, of course, this looks like a proof of principle experiment. To some extent it is, but it's not just that. There's another reason why to do these experiments. These so-called charged nitrogen vacancies currently are very hot candidates for quantum computation, qubits at room temperature, because they have a spin state with a very long coherence time. And also the spin state can pick up magnetic fields, so to speak, at a nanoscale. We have nanosensors in here. But STED is perhaps the best way of reading out the state. And this makes that perhaps an interesting candidate in the end, maybe for reading out, say, qubits in a quantum computer. Who knows? The development goes on. So I'm coming back again to STED. I said the name of the game is on-off. This is how we separate it, a bright state and a dark state. Now, once you realize that this is a general principle, not just doing stimulated emission, then you know, okay, there must also be other on and off states in a dye that you can use to the same effect, do the same thing. Well, and so I browsed other textbooks, and I found there are triplet state, dark states, and of course, if you look into your chemistry textbook, you will find there is cis-trans isomerization, as you can see in here. And then there's other options to do the same thing. You may ask again, why do you use these special transitions that are not found in any fluorophore? You need special fluorophores for this. If you have this basic transition here, that's just totally basic, ground state, excited state. Well, there is a very strong reason for looking into these other types of states. Let me show you the reason now. Look at the lifetimes. Here, the lifetime of the state, the excited state is nanosecond. Here, the lifetimes of the state is microseconds. Here, it's milliseconds. So the lifetime of the states increases. So why is this important? Well, it's very obvious. We separate by having adjacent features in two different states. But if the state, one of the states, disappears after a nanosecond, then this difference in states disappears after a nanosecond. And so, of course, you have to hurry up putting in the photons, creating this difference in states and reading out this difference in states. But if you have more time, microseconds, milliseconds, you can turn it off, read it out, turning on, turning it off. It stays there because it has a long-lived state. So you don't have to hurry up putting in the light. And that makes it, so to speak, operational at lower light levels, at much lower light levels. So to be more formal, this intensity threshold IS scales inversely with the lifetime of the states involved. And the longer the lifetime, the smaller is the IS. So you can break the diffraction barrier using this type of transition at much lower light levels. So the IS, the intensity, threshold intensity goes down from megawatts, kilowatts down to watts per square centimeter, 6 orders of magnitude. That makes this transition, of course, very interesting. Here in the equation, IS goes down, and with that, of course, also I goes down, because you don't need as much light in order to get the same D. Now, this cis-trans isomerization is particularly interesting because it's found in switchable fluorescent proteins. And so we looked into this very early on to check whether we can use it for a STED-like recording. And this is, I called it resolved because I couldn't have called it STED anymore. There's no stimulated emission in it, so I had to give it a different name. Now, the strengths, if you need little light, not only is that you can get high resolution at low light levels, you can say, oh, I'm buying a cheap laser, CW laser, and I spread out the light over a large field of view because I don't need so much. And then I can parallelize the recording, can make many holes at the same time and read out a large field of view quickly. It doesn't matter that I have many. As long as they are further away, that Abbe's diffraction barrier, I can read them out in parallel, project it onto a CCD camera. That's it. Just a few scans in this direction and in the other direction, and I can take a large field of view. And this was done in here. So here, this, say, living cell was recorded within 2 seconds with more than 100,000 donuts, so to speak, in parallel. So again, the key is the molecular transition. So selecting the right molecular transition determines the parameters of imaging. So the microscope and the contrast modality and all the factors are determined actually by the molecular transition that is chosen in here. Okay, then I put up the next question. So what does it take to get the best resolution? Now let's assume you had asked this question in the 20th century. What would have been the answer? I'm sure you know it. Good lenses. Sure, good lenses. Why? Because the separation was done by the focusing of light. And then, of course, you need good lenses to produce the best focus here or the best focus there. And if you cannot produce a smaller focus of light, you're lost. So if you have several features, of course, falling within this diffraction zone, then you cannot do any better. So you're definitely limited by diffraction if you separate by the focusing of light. No way to tell these features apart because everything overlaps in here. So what was the solution to this problem? Not separate just by focusing. Separate by molecular states. In the easiest case, on-off states. Because if you separate by molecular states, you can distinguish the features and can tell it apart, even though they are designed within the diffraction barrier. And now we can tell this one apart from here, from the rest, and can discern it. And you have here a choice of states that I've shown already that you can use to distinguish the features within the diffraction zone. Now, in the methods that I've described, STED, resolved, and so on, the position of the state, where the molecule is on, where the molecule is off, is determined by a pattern of light. You've seen it in the donut. It clearly determines where the molecule has to be on and where it has to be off. X, Y, Z is tightly controlled by an incident pattern of light. I go to the next. Again, I know the position already. I don't need many photons here because everything is determined here by the position of the pattern. Now the question comes up, how does this compare with the seminal invention by Eric Betzig based on the discovery of W.E. Moerner that you can detect single molecules? Well, in the Planck concept, there are two fundamental differences to STED. First of all, it relies on single molecule detection, critically relies on that. That's very clear. Secondly, unlike in the STED case, in the Planck case, the position of the on state is totally uncontrolled, totally stochastic. One molecule pops up somewhere randomly in space. And you create, in this way, this on-off state difference. But since you don't know where you've turned it on, you have to have a pattern of light with which you can find it out. And this is done with a pattern of light that is emitted here by the molecule. Now, without going into the detail, of course, this pattern of light allows you to determine the position with a centroid calculation. The point that I'm making here is you need a bright pattern here to find out the position, just as you need a bright pattern here to determine position. You always need bright patterns of light when it comes to positions. And it's not surprising. If you have just one photon, it goes astray. Here, 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 within the diffraction zone. No way to control where it goes. If you deal with positions, you need many photons by definition, because that's inherent in the diffraction. So, many photons. But the separation, of course, in both cases, is done by an on-off transition. This is how we tell features apart. As a matter of fact, all the super-resolution methods that are in place right now and are useful, they use the separation, they make a separation by transiently placing the molecules in two different states for a time period of detection. Fluorescent and non-fluorescent, that's the easiest to play with. And so this is what has worked out so far. Now you can say, in the 20th century, it was the lenses which were decisive. And the lens makers were the kings. You had to go to them and ask them for best lenses to get the best resolution. But today, no, it's not the lens makers. This is about molecular states. And molecular states is, of course, about molecules. The molecules decide now how good you can image. The molecules decide now the spatial resolution. And molecules, that's not optical technology. That is chemistry. It's now the chemists who make the best pictures in the end. So in a way you can say that this initially physics problem, which diffraction barrier was, no doubt about it, has now been converted into a chemistry topic. And with that, I'm coming to my almost last slide. I couldn't have told you this development without the help of many very talented people who have contributed to this development. Actually, most of them that are shown here are here. I thank them very much from the bottom of my heart for their contributions. And I would like to add that they are still continuing because this field is very, very, very exciting. And I would like to make another point here. Well, the big award now, the Nobel Prize, was awarded to super-resolution fluorescence imaging. But once you have realized that this is the state transition that we have in here, AB need not be on-off in fluorescence, could also be something else, absorption, non-absorption, scattering, non-scattering, spin up, spin down. Then you can also imagine a super-resolution absorption microscope or a super-resolution scattering microscope if you have the right states. And so the story continues, and I'm expecting more of it to come. It has just begun. And this is really my last slide now. You see now, this is Abbe's equation. It was written there for so many years. It cannot explain the fact that we have now a much higher spatial resolution, but we can deal with it very, very easily. We just plug in the square root factor, and now the good news is it goes down to the size of a molecule, so we can have resolution at the molecular scale. Thank you very much.
So our next speaker, our next chemistry laureate for 2014 is W.E. Moerner. He was born in 1953 in Pleasanton, California. He got his PhD in 1982 from Cornell University, and he is now the Harry S. Mosher Professor in Chemistry and Professor by courtesy of Applied Physics at Stanford, California. Welcome on stage.
Thank you. Well, thank you very much. I want to begin by thanking the Nobel Committee for Chemistry and the Royal Swedish Academy of Sciences and the Nobel Foundation for selecting me for this prize. I am truly honored to share the prize with my esteemed colleagues, Stefan Hell and Eric Betzig. For the inspirations for single molecules, which I want to talk about now, and why imaging and photocontrol and so forth become foundations for a different way of super resolution than what Professor Hell talked about, we have to go back to the mid-1980s. When I was inspired by the work of David Wineland and other people to look at single ions in traps in a vacuum, they were able to observe these single ions and do spectroscopy. But why not molecules? Why couldn't you observe a molecule with visible light, a single molecule? Well, of course, there's a bit of a problem, because this famous physicist who built the foundations of quantum mechanics, Erwin Schrödinger, felt that you could never experiment with just one electron or atom or molecule. And he wasn't the only one that thought a single molecule could not be detected. So a key aspect of the early days was getting to the point where we realized that we were able to do it, that it would be possible. And in order to explain that for this broad audience, I need to explain a bit more about spectroscopy. So let's go back to a simple experiment. Let's take this molecule, pteroline, and put it in a transparent host of paraterphenyl, and I'm showing its optical absorption as a function of wavelength. And here's the visible spectrum to sort of calibrate you as to what colors correspond to what wavelength. And if you imagine what's going on at long wavelengths, there's low energy, and so you don't have any transitions. You go to shorter wavelengths, you begin to pump transitions on the molecule. Let's always focus on this lowest transition, or the first one that you come to. If wavelength increases to the right, then because frequency and wavelength are inversely related, then frequency would increase to the left. And the frequency here is about 460 terahertz, 460 times 10 to the 12th cycles per second. A very, very high frequency. 860, even higher over here. Well, now we're interested in doing low temperature experiments because that's where we first observed single molecules. So let's now expand this scale dramatically by a factor of 25 and cool to very low temperatures in liquid helium and see what happens. So here's what you see, and now I've reversed the direction of the scale. Now frequency is increasing to the right and wavelength increasing to the left. And note what happened to our color bar. We're only seeing a tiny part of orange here. You can see a slight change in color, but we've expanded it dramatically. Still, these represent different colors of light pumping the molecules, and what you see is very, very narrow transitions. The molecule absorptions became very narrow. In fact, you can see that there's 4 different origin sites, 4 different inequivalent sites for this molecule in this crystal. Now, what happens if you go even further down? Notice it's only a small range, only 11 terahertz range now being displayed. Let's now blow up the scale even more for this first transition, the lowest transition, by another factor of 1,000. What happens is that the molecule's absorptions get extremely, extremely narrow. Now, I have a sort of a sketch of what's going on over here. The individual molecular absorptions are very, very small, and you don't see just one line. You see a profile that corresponds to different molecules which are in different environments, which have slightly different frequencies. We call this inhomogeneous broadening, and when I was at IBM Research in 1985, we were interested in studying this effect for a number of interesting reasons. You'd like to measure these widths of these individual molecules, and you have to use some tricks. For example, you could use photon echoes as one idea, but we were using spectral hole burning, because in the 70s, Dietrich Haar, Anshel Goroskovsky, and others showed that you could do something called hole burning. Now, what's that? Think of this inhomogeneously broadened line, and now if I irradiate it with a single frequency laser, which is very narrow, I can select just a small group of the molecules, and if I can change them somehow so that they move somewhere else in frequency space, I leave behind a dip, a lowered absorption, which was called a spectral hole. And the idea at IBM then was to use this spectral hole burning idea to achieve optical storage. Optical storage in the frequency domain. You could move to one frequency or another, and at each frequency you could choose 0 or 1. Do I write a hole or not write a hole? And that would be a way of storing digital information in these molecules. Much work was done to develop molecules to do this and methods to do this. But an exciting aspect for me at that time is that at IBM, not only was technology important, but science was also important. And I could ask this fundamental question: what are the ultimate limits to frequency domain optical storage if you use this hole burning idea? In particular, there's a question that came to our minds: is there a spectral noise of some sort that results from statistical number fluctuations or the discreteness of the individual molecules, which would define the smallest possible hole that could be written? Now, let me explain this with a simple analogy. Suppose you now have just 10 boxes and you've got 50 balls, and you want to throw these at these 10 boxes, with all boxes being equally likely. Well, then you might guess, one possibility is that you have exactly 5 molecules in every one of the boxes. But of course, you know that's not what really happens in a real experiment. In fact, what you really get are different numbers of molecules in the different boxes. In other words, the average might be 5, but in each box you get numbers that fluctuate about this average. And so this is the well-known number fluctuation effect that scales as the square root of n. In other words, in this case, the square root of 5 controls what's the variation that you get from box to box that arises from sampling. Well, now think of this as a frequency axis, just like I showed in that inhomogeneously broadened line, and make the inhomogeneously broadened line be extremely broad. And every one of these bins is a bin of width, the homogeneous width of the molecules. So that this basically means, this effect means that there should be some spectral roughness or fine structure on that inhomogeneously broadened line coming from the discreteness of the individual molecules. Here's a sort of a simulation to show you the same idea. At low numbers of molecules, you see a lot of roughness. The roughness seems to go down in relative size as you go to higher and higher concentration. And so this roughness is something that's coming from the discreteness of the number of molecules. But in the mid-1980s, this had not been observed. That's the key point here now. So we decided that we should try to observe this effect. So with Tom Carter, my postdoc, we first found the right material to do it. Pentacene molecules doped into a paraterphenyl crystal. And here's what the data look like. Here's a piece of the inhomogeneously broadened line, and you can see this amazing fine structure. Here's a blown up part of it where I show one measurement and then another measurement later, sometime later, and you see exactly the same structure. This is not noise. It's not something that's changing with time here, but it's a spectral roughness. Now, why is this interesting for single molecules? Well, it turns out that the absorption that you get, let's say for n molecules, scales linearly with the number of molecules, but these fluctuations have an amplitude that scale as the square root of n, as I've described. That's why we call it statistical fine structure. And this detection was achieved with FM spectroscopy, because FM spectroscopy only measures the difference of the absorption from the average value. It directly arises from the discreteness. And in fact, this is what gave us the feeling that a single molecule limit is within reach. In other words, if this absorption is coming from, let's say, 1,000 molecules that are absorbing at each wavelength, then these fluctuations are about the square root of 1,000, or about 32. This signal represents the signal from 32 molecules or so. And that means that our sensitivity of our apparatus only has to be increased by a factor of 32 to get to the single molecule limit. Therefore, it was going to be possible. So that's why Lothar Kador and I used this FM spectroscopy method from Gary Bjorklund to push that method to the single molecule limit, detecting single molecule absorption on the transmission of the light through the sample. Still using pentacene and paraterphenyl. A single molecule absorption looks like a single object there. FM makes a copy of the in 2 places, and another secondary modulation that we used takes a derivative of this, so the single molecule spectrum should look like a little W, and here you can see it in our samples. Here's no molecules and many molecules. Now, this was, of course, the hard way to do this experiment, but nevertheless, it had some interesting properties. It's insensitive to scattering. It is limited by laser shot noise. And, of course, the challenge was, they made it hard, is that the focused laser intensity had to be kept very low, because if you broaden the molecules, then there's amplitude of these effects gets smaller and smaller. But at least it proved that a single molecule could be observed with visible light. The FM technique is one in which you take the laser and frequency modulate it. So here's frequency modulated light, here's the sample, and the sample converts this constant amplitude beam into an amplitude modulated beam. That's what you end up detecting. It's very much like FM radio at 506 terahertz, because that's the center frequency now of this huge inhomogeneously broadened line. Well, a year later, a truly important advance occurred, because Michel Horry showed that you could also detect single molecule absorption using the emitted fluorescence. Here's some of his data. Here's an inhomogeneously broadened line, and these little dots are the individual molecules. This is the same system, inosine and paraterphenyl, but now you have a method that is sensitive to scattering from the sample. So these samples had to be incredibly clear and perfect, crystal clear, sublimed flakes so that you don't produce scattering backgrounds. Any sort of scattering background can make it harder to see the molecules, but that's possible to overcome. And so these methods produced higher signal-to-noise ratio for similar bandwidths. So essentially everyone switched to this fluorescence method after that. Well, here's some examples of single molecules at low temperatures from Pat Ambrose's work in my lab. Here's the inhomogeneously Broughton line, and if you blow it up, you see these beautiful spikes, and in looking at just one, you see a width of about 7.6 megahertz in the visible, where the center of this is, as I said, 506 terahertz. That's an incredible resolution, spectral resolution of a factor of about 50 million improvement. And that's why at this time we weren't really thinking a lot about spatial resolution, because we had such beautiful spectral resolution and could explore many properties. Nevertheless, we did take some images of these single molecules. And here's one where this is the frequency axis where we're tuning the laser, and this is a spatial axis where we're moving the laser spot over the molecule. And you can see they make little mountains in this three-dimensional representation. And we knew at that time that the width of the peaks along this position axis was basically a direct measure of the intensity profile of the laser beam inside the sample. In other words, you take this nanometer-sized molecule and move it over the laser beam spot, and you can measure its shape. That's very similar to what gets done for super resolution. I'll talk about in a few moments. Also in those years, in the early 90s, in Urs Wildt's lab, a wide field two-dimensional microscopy was done and to image these single molecules at low temperatures. Well, if there were just images and just molecules that sat there or whatever, of course it's exciting to get to this new regime, to see single molecules. But what happened, which was even more exciting, is that surprises began to appear. Here I'm going to show some images, some spectra where the laser is scanning over 400 megahertz, looking at that single molecule, and look what happened. The molecule is jumping around. It's moving from one color to another. That is, even at liquid helium temperatures in a crystal, the molecule is jiggling and jumping from one frequency to another. It was amazing and really exciting to see this when Pat brought the data in and told me about this in the lab. And Jim Skinner helped with a theoretical understanding of this effect, that it's coming from fluctuations of two-level systems inside the crystal near the molecule. And when the nearby crystal changes, then the molecule moves to a different frequency. One of the cool surprises that if you imagine putting a laser at a fixed frequency, then you'll see the molecule turning on and off. You'll see it blinking on and off as it moves into and out of resonance. Another key thing that occurred, Thomas Pache, another postdoc, looked at perilene and polyethylene, and he observed that you could optically change the positions of the molecules. Here's a molecule that's stable at that frequency, and then if you bring the laser into resonance with it, you can make it disappear. It's jumped somewhere else. You wait a little bit, and it's come back, and you can observe it again and again, and do this many, many times, on and off. So this is the early days, of course, of what you would call blinking and switching effects that end up to be useful later. Well, there are examples of why we were so excited about seeing single molecules in those days. We're in a complex system, and that's why removing the ensemble average is very important. You can then explore heterogeneity, see how the different molecules behave differently from one another. You can follow state changes in time and test theoretical effects. The molecule is a nanometer-sized object, so it senses nanometer-scale interactions, and it led to super-resolution imaging, as I'll describe in a moment. There's also commercial applications. Okay, so now let's jump to room temperature. I want to talk about a different thread of work that was going on during the 90s. Forgive me for this list, but I just want to mention some of the key advances that occurred. In the early 90s, 1990, several researchers looked at and saw signals from one fluorophore by measuring the autocorrelation signal from the sample, and single bursts were observed as well in 1990 for just a molecule in solution resolution floating through a laser focus. Later, an important step was imaging a single fluorophore using near-field methods, which Eric Betzig achieved and we'll talk about. And then after that, several other groups also used near-field methods. Confocal imaging followed, then wide-field imaging followed, where you look at, in vitro, look at motors on an actin filament, Janelia, and on the cell membrane, single molecules moving around, single lipids moving around. So there were a number of wonderful advances that caused us all to say, let's move to room temperature, where we can look at a lot of new biological effects and other chemical effects as well. So for all of those methods at room temperature, they roughly work like this for seeing a single molecule. You pump a transition from the ground state to the excited state and collect this emitted fluorescence. And so here's sort of a schematic of a sample where I'm showing that you use these kinds of molecules now, let's say rhodamines or other kinds of Cy3 dyes or even fluorescent proteins, and you often have them attached to the object of interest, and you want to be able to observe singles. Here's the focused laser spot, however, which is limited by diffraction to being only about λ/2 in width. And so that means that in order to make sure you look at only a single molecule, you have to dilute the molecules dramatically to have them be farther apart than the size of this effective focus spot to see single molecules. But in spite of that, much has been done and continues to be done with single molecules that are isolated. For example, I'll just show you very quickly a couple of them. Here is a cell, a CHO cell, and some transmembrane proteins have been labeled with a fluorophore. And if you watch this movie as a function of time, you see something really fantastic. The dance of molecules on the surface of that cell. This is what's going on in the cells in your body right now. The labels don't last forever, of course. You see them turning off. But the little tiny spots are telling us how the motion of these transmembrane proteins occurs on the surface. That's a diffusion effect that can be studied. Another kind of experiment can be done if you do time-lapse imaging. And here I'm showing the Colobacter crescentus bacterium, which is very small, only about a couple of microns long and 500 nanometers across. But So-Yeon Kim observed a particular protein called MREB, labeled with yellow fluorescent protein. And what do you see on a time-lapse, long time scale? You see that this molecule is moving across the cell and then back around the backside. It's moving in a line. This is not at all diffusion like we saw before, but it's a completely different process, and this turns out to have something to do with the formation of the cell wall in these bacteria. Here are the tracks of many single molecules showing you how that occurs. And then the third example that I'm giving quickly is a material science example. In a crystal at room temperature, here you see two different kinds of molecules. Some that are completely fixed, so the spots are not moving, and other molecules that are moving around like flies. They're actually doing that because they're moving in the cracks of the crystal. The moving molecules tell you where the defects are in the crystal. So you want to get the picture now that by watching these beautiful nanoscale light sources, they're very much like the stars in the night sky that tell us something, but here in living bacteria and in living cells, we can learn much more about the dynamics. Well, now let's go to the main thread of this whole Nobel Prize, which is super resolution microscopy. And I want to summarize how you can use these single molecule labels to surpass the Abbe diffraction limit. The answer to that, and stated simply, is that you first have to be able to image these single molecules like I showed you, but then you have to add to that two key ideas. So the first key idea is called superlocalization. In the case of superlocalization, you can think about it in this way. Here is a little cinder cone mountain in the middle of Crater Lake in Oregon in the United States. And that scale bar there is 120 times 10 to the 9th nanometers. So this is human scale, of course. You know that if you take your cell phone, which has GPS capability, and walk to the top of this mountain, you can determine the position of the center of the mountain to very, very high precision. That's the basic idea for determining superlocalizing single molecules. Find the position by fitting the shape of the single molecule image. Illustrating that, here are some of these single molecule images, which are, they're all diffraction limited. That's why the blob has a size of about λ/2. But if you blow up the image on your camera so that multiple pixels sample it, then this cross-section shows you that there's a shape to that blob. It's not just a disk. Some positions on the detector will count more photons, others less, and so that means that you can use a fitting function, in this case a Gaussian function, or more formally an Airy function, to fit that shape. The width of this is, of course, diffraction limited, but if the center position of it, the parameter of the fit that corresponds to where it's located, can be measured to much higher precision than the width. In fact, the estimates of the position of the center have this narrow green distribution with standard deviation sigma, and sigma is roughly given by the Abbe diffraction limit that we've been trying to beat, divided by the square root of the number of photons detected. As more and more photons are detected, you get better and better information about this shape so that you can find the position much better. For example, for 10 to the 2 photons, 100 photons, you can get down to 20 nanometer precision or so. Well, that works great, of course, for separated molecules and has been used a lot in biology. Here's a number of examples of very early superlocalization experiments where LDL particles or kinesin motors and so forth were imaged by finding a large object. But if you find the position of a single fluorophore, you can do something much better, because all the photons come from the same nanometer-sized object. And that means that you can then extract nanometer scale position information about where the molecule is. Watch this little demonstration in a simulation to see what I'm talking about here. Here's the image of the single molecule in time, and you see it's moving around, but each one of those shapes gets fit by a function, and therefore the error in the position is this green curve again. Here's the actual positions of the molecule as a function of time. We can measure these tracks beautifully now inside cells, for example. Well, the key point number 2 is that you want to go to super resolution. That is, you want to resolve 2 objects that are close together. And a key important paper was published in 1995 from Eric, where he suggested using an additional control variable to separate these diffraction-limited spots in the spatial dimension, and mentioned this spectral tunability that was available from the low-temperature single-molecule experiments. And that was actually then demonstrated by a group a couple of years later. A number of groups used several methods to try to separate molecules, either fluorescence lifetime or overlapping, or photo bleaching, or differently colored probes and blinking and so forth. But these various early demonstrations only allowed you to separate a handful of molecules. What was needed was a real general method. And so that gives me key idea number 2. And I'd like to describe it in this general way by calling it active control of the emitter concentration and sequential imaging. So here's the idea. We want to observe this structure, and we decorate it with these labels that are fluorescent labels. Now, the problem is if you let all of them emit at the same time, then each one of those diffraction-limited spots will all overlap, and so you don't see the fine structure. So the solution is simply don't let them emit all at the same time. Use some sort of active mechanism that you have control over as the experimenter to keep the concentration low at any time. For example, let's suppose that we have a photoactivatable fluorophore that's dark all the time unless you turn it on with another control beam. Then you can do that at very low intensity and turn on only a few of them and localize those individuals. Then you photo bleach and do it again. You'll get different samples of the underlying structure. And from these super localization positions that you record, then you put them all together in a pointillist-like reconstruction of the underlying structure. So that's the idea, breaking the diffraction limit using single molecules as tiny light sources. This idea appeared on the scene, and I heard it first from Eric Betzig in April 2006 when he described PALM at an NIH meeting in Washington. And quickly, STORM appears, FPALM appears, PAINT, DSTORM, all kinds of people showed that different mechanisms could be used for this active control idea. So it was a really incredibly exciting time to hear about all that. We did something called YFP reactivation then, and we didn't have an acronym for it, of course, so it was forgotten, right? So here's a new acronym I can add onto the pile that's mechanism independent, single molecule active control microscopy, or SMACM. So that's just for fun. I'm glad that you're laughing about it. Now, YFP reactivation, let's talk about that for a moment. Because when I heard about all these exciting developments, I said, well, how can we do this? What active control mechanism could we use? And I remembered this experiment that we did back in 1997. Rob Dixon was looking at and wanting with me to achieve the first imaging of a single fluorescent protein. We got some enhanced yellow fluorescent protein from Roger Chen, and here's one of the images that were recorded of the single molecule. But as before, just like at low temperature, when you first see a single molecule here at room temperature of a fluorescent protein, surprises might appear, and they definitely did. We saw that the fluorescent proteins were blinking. They would emit for a while and then turn off and turn back on again and turn off and turn back on again. This means that while we're getting a lot of photons in this emissive state, the protein goes into a dark state and turns off, and then thermally comes back and starts emitting again. Rob also saw that if he radiates the molecule for a long time, you go into a long-lived dark state, so it seems like it's completely photo-bleached. But if you use a little blue light, you can turn the fluorophore back on and observe it again for a long time until it finally goes into a long-lived dark state. A light-induced recovery of the molecules, another way to control the emission of single molecules that can be used for super resolution. These sorts of ideas, of course, spawned an industry of further development of switchable fluorescent proteins. Photoactive GFP, you'll probably hear about later. DRONPA and other sorts of fluorescent proteins were created where you try to optimize the switching mechanisms. What do the data look like from this blinking process in YFP? So here's an example. I show here some bacterial cells, and this is the white light image of the bacteria. And here's the fluorescence image, and now here's the movie. This is single YFP fused to a particular protein inside these bacterial cells, and you see all these little blinking stars that are coming from just inside each of these cells. That's so wonderfully amazing sort of kind of imaging that allows you to, in each frame of the movie, fit the positions of the molecules and then look at the structure of the underlying proteins in the bacteria. How about some additional examples? Let me show you. In a collaboration that's been going on in my lab with Lucy Shapiro for some years, we've been looking at Colobacter crescentus, an interesting bacterium. And here's 3 different proteins. Each case has been fused to YFP, MREB, PAR-A, and HU. These are the diffraction-limited images. And these are the ones that led people to say, oh, well, light microscopy won't be useful because it doesn't have enough resolution. But with these super resolution methods that I've described, this is what you see. This is, of course, the eureka moment that we've had when you are able to look inside a very, very tiny object and see far more detail and learn that even in these bacteria, there are structures that are helping to control the behavior and the development of this organism. This particular protein binds to DNA, so all of these bright spots are coming from the positions of the DNA inside the bacterial cell. So that's examples of using fluorescent proteins. What about using other methods of achieving super resolution? For example, that paint method. I'll describe it very quickly here. In this example, there is a cell off the screen, which is a PC12 cell, which is a neuronal-like cell, a model cell for neurons. And this is an axonal-like projection that comes out from the cell. And the way we're observing the system is to use this beautiful molecule that synthesized by people in Justin Dubois' lab, a molecule called saxitoxin, which is a toxin that binds to voltage-gated sodium channels. He can make it be fluorescent, so each of these spots is coming from this labeled saxitoxin as it binds to voltage-gated sodium channels in the membrane of this axonal projection. So remember, what's going on here is that in solution, these molecules are all floating around and they're dark because they're moving around. But when they bind to the surface, you see the bright spot clearly and can localize it and record it as part of the super resolution image. The beauty of this particular experiment, since it's on a live cell, is that you can, as a function of time, observe how the cell changes. This particular display, I hope that you can see it clearly without too much washout by the lights, shows that each of these spots coming from voltage-gated sodium channels allows us to see how the neuron is changing with time. There are new neuritic projections that start appearing and disappearing during the time that this piece of the cell has been observed. The time resolution here is about 500 milliseconds. Okay. As one final example, I want to talk a little bit about the frontiers in this area. We have new fluorophore. We need new fluorophores, and we need new imaging methods. Let's have some quick examples of that. In the case of fluorophores, here's a particular interesting molecule called a rhodamine spirolactam that has a closed form where the molecule is off and an open form where the molecule is fluorescent. And by modifying the structure of the molecule using friends in synthetic chemistry from Bob Twiggs' lab, we can make this molecule switch with blue light so that you don't damage the cells as much. If you use this functionality to attach the molecule to the surface of the cell, this will bind to amines on the surface of a cell. Here are Caulobacter crescentus cells, where you can see the super resolution reconstructions that show you clearly the subdiffraction stalks that are present on these cells that you don't see by normal microscopy. So that's just showing that there's great work to be done to develop, using chemistry, new molecules that can be used for these photo-switchable and photo-controllable mechanisms. In terms of methods, there's been a lot of interest in 3D methods. We've used this particular one where we convert the standard microscope into what we call a double helix microscope. The standard microscope, as you move in Z, you see a single molecule going in focus, out of focus, in focus, out of focus, and so forth. But with the double helix, the single molecule looks like two spots that revolve around one another. So I just, for a particular position Z, I just look at this angle of the two spots and I can tell you what Z is. All right? So you get X, Y, and Z from the camera, object depth being encoded into this revolution of the two spots. And this is what you see then for Caulobacter crescentus cells labeled on the surface with this molecule, but imaged in 3 dimensions using the double helix microscope. So what's so exciting about all of this work that's been going on is that it's had impact over many areas of science, in chemistry and physics and in biology. And this idea of working at the single molecule limit, this ultimate limit of individual emitters, has attracted the attention of many talented scientists who make lots of contributions. And I've listed more here, but there's no way I can possibly describe all the exciting things that are going on. And what's particularly exciting is that you can use these molecules now for going beyond the optical diffraction limit. So I have a lot of people to thank for making it possible for me to be here. This is thanks to the Moerner Lab alumni from IBM Almaden Research Center, from UC San Diego, and from Stanford University. But I also want to thank my current guacamole team. Here we are at Halloween, sort of dressed up a little bit in some funny way. We have this logo that says no ensemble averaging. Of course, what is this guacamole, right? What is that? Well, of course, one molecule is one guacamole. That is 1 over avocado's number of moles. So this is such a bad pun. I have to apologize to the memory of Amadeo Avogadro. I also want to thank my collaborators and colleagues at many locations, from WashU to Cornell to IBM to ETH, Zurich, UCSD, and Stanford. It's been truly wonderful working with all of them. I want to thank my mentors and the locations and the funding sources. Here's a couple of the key mentors. Here's Jim Miller from Washington University, my undergraduate institution, and Al Sievers from Cornell University, my graduate institution. But there were many professional mentors. I thank funding agencies who made a lot of this possible, and the support from all these institutions. And finally, I want to give thanks to my family and friends. Here's my stepmother, Esther Moerner, but here are my parents, who were the ones who taught me to love science and taught me to figure out how things work and how to fix things, which really made me an experimental scientist. And I, of course, owe everything to them. But also, I want to thank my son and my lovely wife, Sharon, who has been giving me a tremendous amount of support for more than 31 years. Thank you very much for your attention. Not now.
Now, the third chemistry laureate for 2014 is Eric Betzig. He was born in 1960 in Ann Arbor. He got his PhD from Cornell University, Ithaca, New York, and he is now the group leader at Janelia Research Campus, Howard Hughes Medical Institute, Ashburn, USA. Please come onto the stage.
One of the nicest things about winning the Nobel Prize is hearing from all of the people in your past and having the time to reflect on the important role they've each played in getting me to this very happy and fulfilling life I have now. So to all of my friends from grade school up to my peers who nominated me for this honor, you have my deepest thanks. So, let's see. So I got started in super resolution back in 1982 when I went to Cornell University for graduate school and met my eventual thesis advisors, Mike Isaacson and Aaron Lewis. So Mike had recently developed a means of using electron beams to fabricate holes in a membrane as small as 30 nanometers and then make the membrane opaque. And he and Aaron figured that if we could shine light on one side of the screen, then the light that initially comes out the hole from the other side would create a sub-wavelength light source that you could then scan point by point over the sample and generate a super resolution image. And so the idea was to try to create an optical microscope that could look at living cells with the resolution of an electron microscope. Compared to that, everything else in the department seemed boring to me, so I said, please sign me up, because that sounded really exciting. So at that time, many people told us that this idea would never work, either because it violated Abbe's law, or even worse, the uncertainty principle. But all doubt was removed from my mind in '84 when we learned about the work of Eric Ash and Nichols, who in 1972 used 3-centimeter microwaves and was able to, by near-field techniques, get 1/60th of the wavelength of resolution of light in test patterns in a beautiful paper in Nature. And in fact, the idea for near field goes back even farther, originally to E.H. Singe back in 1928, and many, many people have independently come up with the idea since. For far field super resolution, the earliest demonstration I'm aware of is by Lukos. So back in 1967, by inserting grating masks at planes conjugate to the sample and the object, he was able to beat Abbe's limit by about a factor of 3. But this is at very low numerical aperture, but still a demonstration that Abbe's law was not inviolate. And in fact, far-field super-resolution has a very long history, particularly in the semiconductor business, where the nonlinear interaction of the light with photoresist has been a staple of making line widths far smaller than the wavelength of light for a generation. Even more impressive, though, is how visible light is used to inspect semiconductor wafers, and by having a priori knowledge of the pattern you actually expect to create and doing theory about the diffraction of light from those patterns and comparing the actual data you get against that theory, people in today are able to measure and see the line widths of these features to about 1,000th of the wavelength of light. So really, at some level, super resolution is not new at all, and there's people in Silicon Valley who are probably laughing at us here today, thinking that we're the guys who invented this, when this has been a staple for a very long time. But in my mind, really, the guy who deserves the lion's share of the credit for not just pushing a little bit beyond Abbe's law of lambda over 2n, which is still 2n sine theta, which is still related to numerical aperture, but actually breaking through the more theoretically supposedly insurmountable limit of lambda over 2 is Eric Ash, because he was the guy who really shattered it by getting to lambda over 60 with the near field technique. Speaking of shattering, the probes, the types of apertures we were making in those thin membranes would break all the time. They were hard to make. They were costly. So eventually we abandoned that, and we instead pulled glass micropipettes, similar to the method that was developed just a few years before in patch clamping for single ion channel recording. We would then coat these with aluminum to create an opaque structure, except for the little hole at the end that would then be our aperture. So with that, I built this monstrosity you see here, which was my first near-field optical microscope. I cringe now at how complex and crazy this thing was, but at least it gave me the ability to learn the system engineering skills I would need to become a true engineering physicist. And eventually I was able to surpass the diffraction limit with this near-field microscope that I did in my thesis. So that microscope was frankly a pain in the ass to work with, and the resolution gain was at best about a factor of 2 beyond Abbe's limit. But it was good enough to get me my dream job at Bell Labs. And so I started trying to develop that technique further for a few years, and it was a very hard slog. But thanks to the patience and encouragement of my boss, Horst Stormer, I eventually came to realize that that probe was not a really good design because the light that was sent down that taper was largely retroreflected back before it ever got to the tip, and the little bit of light that did make it to the tip were in electromagnetic modes that didn't couple well to the aperture. So postdoc Jay Troutman and I then instead created a probe that consisted of an adiabatically tapered optical fiber which would guide the light very efficiently to the tip region and then efficiently couple that light to the evanescent modes in the aperture. This made a probe that was 10,000 times brighter than the earlier probes and also then allowed us to routinely get to about 50 nanometer resolution. Part of that was I also, in the same year, invented a means to dither the probe back and forth, oscillate it, and then as it would come close to the surface, that oscillation would be damped, and by that I could regulate the distance of the tip from the sample. So with these two innovations, near-field became fairly routine. In 1992, we had the world record for data storage density when we could read and write bits in magneto-optic materials as small as 60 nanometers. We also demonstrated super-resolution photolithography, nanoscale spectroscopy, and applied refractive index, absorption, and polarization contrast. In fact, to this day, near-field remains the only diffraction unlimited technique which can use the full panoply of optical contrast mechanisms and isn't dependent on, say, a switching mechanism in fluorescence. But the mechanism that's probably most important for biology is fluorescence because it offers protein-specific contrast. In '93, we were the first to demonstrate super-resolution fluorescence imaging of cells when we looked at the actin cytoskeleton in fibroblast cells. What was impressive about this, though, is that what we could tell from the signal noise that we had on these single actin filaments and the size of our aperture was that it should be possible to see single fluorescent molecules. This was a very hot topic at the time because just a few years previously, W.E. and Michel Orit had broken to this ultimate level of sensitivity of single molecule detection at cryogenic temperatures, and several groups such as Dick Keller at Los Alamos and then Rudolf Riegler had already shown at room temperature in solution that you could see bursts of fluorescence from single molecules. Key to these later experiments was the idea that you had to restrict the excitation volume to reduce the background. Well, that's what near-field excels at, is keeping a small excitation volume. So as soon as Rob Chichester and I decided to try to look at single molecules, on our very first try, we got really great results. And this is some of this. But the weird thing was, instead of seeing a bunch of just blobs or round spots, they would instead look like these crazy arcs or ellipses or other things, and these would change as we changed the polarization of the light. So I still remember running excitedly to Horst's office and trying to understand this, and together with his help, realizing that what we were seeing was the interaction of the electric dipole moment with the evanescent fields inside of the near-field aperture, and that was what was giving rise to these patterns. And so what that means is that we could actually turn the experiment around and think of the molecule as the light source and the aperture as the sample. And by choosing molecules that were oriented along the X, Y, and Z axes, actually map out the nanoscopic electric fields inside of that aperture. We then compared this to a theory that Hans Bethe had developed back in 1944 and was able to show very good agreement. So once we had that, then we could use Beta's model to predict what kind of pattern we would see for any orientation of molecule, compare that to our data, and hence find the dipole orientation of every molecule in the field of view. And given that information, we were then able to fit these crazy shapes to the theory and find the positions of these molecules down to about 12 nanometers in X and Y and about 6 nanometers in Z. So this became very influential for what was to happen later. So in another pivotal experiment, I joined forces with my best friend and colleague at Bell, Harold Hess. So Harold had made a name for himself at Bell a few years earlier by building a world-class scanning tunneling microscope with which, among other things, he discovered the core states at the centers of these vortices in the Abrikosov flux lattice of type II superconductors. Harold's and my interest was to combine my near-field probe with his low-temperature scope to be able to study excitons, which are the sources of light generation in semiconductor heterostructures, such as in this laser pointer and such as that won the Nobel Prize in 2000. So the goal was then to combine the high spatial resolution with my near-field probes with the high spectral resolution we could get in Harold's rig by running near absolute zero. So when we did this, we were surprised to find that the normally smooth spectrum that you see instead would break up into these crazy sharp lines. And furthermore, as we drove the probe even small distances from point to point, this spectrum would change completely. And what we eventually realized is that we were seeing that the excitons could not emit anywhere, but there were only certain specific points of exciton recombination, and the colors or where these points were was based on the local thickness of the quantum well at that particular spot. And so what was probably more important later on was that even though there might be a dozen or more of these emitting sites underneath even the small size of the near-field probe, we could still study them individually because they glowed in different wavelengths. So if we built up this higher dimensional space of X, Y, and lambda, then we could look at them. So this is now then coming up to about 1994. And at that time, the limitations of the near-field technique became incredibly obvious. The foremost of these is that the near field is ridiculously short. And so some of these problems are just engineering challenges, but some were truly fundamental. And so it was clear because of that short depth of focus, there was no way I was going to realize my ultimate dream of looking at live cells with the resolution of an electron microscope. So I got very frustrated. At the same time, though, near field got to be a big fad, and like all scientific fads, you have a lot of people jumping in the field. They do sloppy results. They push all the problems underneath the rug, and they overhype its capabilities. And so all of that made me very uncomfortable. And the third thing that tipped the balance for me was Bell, you had to work really hard to succeed at Bell. But what was happening by '94 is you could sense the changes that were happening in the company and that they would no longer value basic science in the way they used to. So it took these two young and innocent guys like this in '89 and turned us into these sort of stressed and worn out guys you see there just 5 years later. So all of that combined, all three of those things, and I said, screw it, I'm sick of science, I really hate academia, I quit. And so I just quit. And so I really had no idea what I was going to do next, but after a few months of trying to flush near-field microscopy out of my head, I was walking my daughter around in a stroller, and it hit me, I don't know how or where from, that you could combine that single molecule experiment I did with the spectroscopy experiment Harold and I did to come up with a different far-field way of doing super-resolution imaging. So the idea is if you have a bunch of molecules that are so close together the diffraction limit spots overlap, we've already heard about this, if you can find some way in which they differ from one another, and it can be anything, then you can isolate them in a higher dimensional space. But once they're isolated, you can find the centers of each one of those diffraction limited spots to much better than the width of the spot, and hence you plot all the coordinates of the molecules. So I published that idea in 95. In 99, Van Ooyen and colleagues first demonstrated this by spectral isolation at low temperature and found 7 molecules in one diffractional limited spot, find them in 3D. And then in the 2000s, several groups extended this to room temperature. But again, by different means, photobleaching, lifetime, and blinking. And this was really a general concept I was trying to get across here. The problem with all of these methods, though, is based on something called the Nyquist criterion. That if you want to make any microscope image of a certain resolution, you have to sample the resolution element divided by 2. If I only go once every half period of this pattern, I can miss it completely. And so what that means is you have to have the ability by this method to see one molecule in 2 dimensions if I want to get 20 nanometer resolution on top of 500 that would be glowing at the same time. And none of the methods I just described were at the point of having that much isolation in that 3rd dimension to get very much beyond the diffraction limit. I didn't have a really good idea in 95 other than running at cryogenic temperatures with a near-field microscope. That was going to be a hero experiment, and I was sick of science, so I just published the idea and left it at that. Eventually, I ended up working for my dad's machine tool company in Michigan, where I did a number of things, but this was my best baby, the one I'm proudest of. This was a servo-hydraulic machine tool that would use an old hydraulic technology, marry it to sort of energy storage principles that you have in hybrid cars today. It would move 4 tons at HEs of acceleration and position it to 5 microns, collapse the size of a machine tool from the size of the stage to something, much smaller, and be much cheaper, much faster, much better. I spent 4 years developing that idea, 3 years trying to sell it, and in the end I sold 2. So what I learned is that I may be a bad scientist, but man, am I a worse businessman. So by 2002, I said, Dad, I'm tired of wasting your money. I'm sorry, this just isn't going to work. And so I quit and had no plan B as usual. And so this was then the darkest time in my life, right? Because not only had I pissed away my academic career, I had also blown up my backup plan of following in my dad's footsteps. I'm 42 years old with 2 young kids and no job and no prospect of a job. So, but I did something smart, is that Harold had also gone into industry where he was considerably more successful than I was, working for a startup in San Diego. So I reconnected with him, and we just started getting together in different parts of the country, the national parks, and just, talking what's the meaning of life. How can we have an impact before we die? What's interesting? What we realized is that while neither one of us fits well in the normal academic scheme of things, we both really love science and we love the ability to be able to pursue our curiosity. So we started trying to think about what we could do to have an impact in science again. But as you can see from these pictures, is that, as always, Harold is always 3 steps ahead of me. So that caused me to start reading the scientific literature, which I hadn't done for 10 years. And the first thing I ran across was green fluorescent protein. And it was a revelation to me, a shock, because it was such a big problem in the near-field days, the labeling of cells to get it to work at high enough density and specificity. And the notion that you could coax a cell with a little bit of jellyfish DNA to be able to get it to produce any protein you want with a fluorescent tag on it. Just, my jaw was hanging down for a week in astonishment at this. And so when I was casting around for an idea, I didn't want to do microscopy, but as soon as I saw this, I said, oh shit, I got to do microscopy again. So I said, okay, microscopy it is. And so Harold and I continued to look around. But while I was taking my holiday from science, science wasn't standing still. So right after GFP, a lot of people want to understand the photophysics of that protein, in part to be able to do mutagenesis to get different colors so they could do multicolor imaging. So Stephen Boxer in '96 noticed that there isn't just one absorption hump for GFP, but two. And what was even crazier is that if you would study at this near UV peak for a while, it would go down, but the peak at 488 would go up, that there was some kind of weird photoactivation effect in GFP happening. Then Tobias Meyer's group actually exploited this for what was the first sort of photoactivated pulse chase experiment, where they used wild-type GFP and used UV light to then turn on GFP in a certain part of the cell to then watch how those proteins go to other parts of the cell. The following year, WE was able to show this same phenomenon with Rob Dixon in GFP at the single molecule level. So then George Patterson in about 2000, he was in Jennifer Lippincott-Schwartz's group at NIH, was very interested in following what Tobias had done. The problem was that the on-off contrast ratio for wild-type GFP was very low. So he did directed mutagenesis and eventually came up with what was called PA-GFP, where you could turn on the fluorescence of molecules with a much higher contrast ratio and use these in much better pulse chase experiments. So in 2005, Harold recommended that we go visit the National High Magnetic Field Lab that was headed by our buddy from Bell, Greg Bobinger, so that we could meet this guy, Mike Davidson. Mike was a microscopist who had made a fortune selling neckties that were emblazoned with photomicrographs of cocktail mixes. And he channeled that money into creating the website tutorials for the major microscope companies, made a lot of money from that, and then used that to follow his passion of doing live cell imaging. And he eventually developed a library of 3,500 different fluorescent protein fusions. When we visited Mike, Harold and I first learned about photoactivated GFP and the other photoactivated proteins that had come along. And so I vividly remember Harold and I sitting in the airport at Tallahassee, and then both of us being thunderstruck when we realized that this idea of being able to turn on molecules at one at a time was the missing link to make that idea I had pitched 10 years earlier to work. And so I had been pursuing another microscope idea at the time. We dropped that like a hot potato, and we said, this is easy, let's do this and do it now. The problem is that Harold had quit his job a few months before, so now you have two guys who are unemployed, and how the hell are we going to do this? It's going to take too long to get a grant, too long to get VC funding. So because Harold doesn't burn his bridges as effectively as I do, he was able to take a lot of his equipment from Bell. And so we pulled that out of the storage shed, put $25K each of our own money into it. And normally you would do it in the garage like Jobs and Wozniak, but we were able to put it together in Harold's living room because he wasn't married. So there was nobody in the way to prevent that from happening. But we knew we had to work fast because this idea was going to be ripe and in the air. So we worked around the clock day and night in order to do this, or at least Harold worked day and night. I found the couch sometimes too comfortable, and so Harold would tease me and keep taking pictures of me while I was asleep. But we were missing one thing, is you had two physicists who were totally naive about biology. And so we needed a good partner in that regard. So when I was pitching this other microscope idea, I called a buddy, Rob Tico, who had also been at Bell and was now at NIH. And so I was going to pitch that microscope, and that's the talk I gave. But I said, when I'm there, would I please, please, please be able to meet George Patterson and Jennifer Lippincott-Schwartz? And so I took George and Jennifer to lunch, and I swore them to secrecy and told them the idea that Harold and I were working on. Many people would have blown us off because we were two crazy guys who hadn't published a paper in 10 years. Jennifer doesn't think that way, and I owe a lot of my success to her as well as Harold. She said, sure, bring it here. So we packed up the instrument from Harold's living room, and actually moved down in scale to actually working in this dark room in Jennifer's place, which is a lot less comfortable than the living room. But then started doing experiments. And very quickly, when we brought it in, and then George did all the cell culture and transfections and the molecular biology to try out a whole different number of protein fusions. And we got to the point where you could turn down the violet light so low that a few molecules at a time would come on. And then if you sum up those spots, you get the diffraction limited image, but instead, if you find the centers, you start building the palm image. And after 20,000 frames, you go from the diffraction limited image to the super resolution image, or to appreciate better. So with high enough labeling density, you can get down to 20 nanometer resolution in your living room by this technique. So it's a fairly simple method. So in a way, though, Harold and I got lucky, because we got lucky that there were certain photoactivated proteins and cage dyes that had enormous on-off contrast ratios. There's a lot of work in this field where I still don't feel like the field has really appreciated how important that on-off contrast ratio is to get from smushy-looking results like that to much crisper results because of the background problems that you face. So again, 2005 was the luckiest year of my life, not only for doing PALM. In PALM, we went from the idea to having the data for our science paper that got me on this stage today in 6 months. And that's what we could do because we were working alone in a living room, which is a very effective environment. So, but in the same year, by a different crazy set of circumstances, I got introduced to this guy, Jerry Rubin. HHMI was starting to want to build a freestanding research institution modeled on Bell Labs. And so the rebirth of Bell Labs caught Harold's and my interest. This guy's farsighted enough to hire 2 guys who hadn't published a paper in 10 years. This was before the Palm paper came out. And so we went from rags to riches. Once the institution opened, we went and ended up building next generation scopes. I hired a talented guy, Hari. Harold hired Gleb, and then we did this type of work. So in my group, we focused on applications for the first few years. Here with Jan Liphart's group at Berkeley, we looked at chemotaxis receptors in E. coli, and we're able to show that these various cluster sizes you see in their positions along the poles are completely predictable in terms of a stochastic model of self-assembly where the proteins are randomly inserted in the membrane and then diffuse until they stick to an existing cluster. We also showed that many proteins, such as in these focal adhesions that attach the cell to the substrate, which look co-localized at the diffraction limit, are nothing like co-localized at higher resolution. With Bob Tejan's group at Janelia, we're able to show a mechanism of gene silencing where the core promoters are spatially segregated from genes that hug up against the nuclear membrane. And with Tom Blandpede's group at Maryland, we were able to look at live cultured neurons and show that the actin that gives shapes to the dendritic spines is only polymerized in certain discrete locations. At the same time, Harold, being the better physicist than I am, built the ultimate palm microscope that used a 3-phase interferometer concept he originally developed in industry to measure the fly height of recording heads over recording discs, but with that has even better sensitivity in Z than in XY, worked with Claire Waterman to unravel the entire architecture of focal adhesion proteins vertically from the substrate up to the actin cytoskeleton. In a recent paper with Jennifer's group, they were able to resolve an unknown question about escort proteins which are involved in HIV budding as to whether these act outside of the bud or inside of the bud, and they showed that the latter is true. Harold has also worked, because he has a lot of background from industry in electron microscopy, at correlating electron microscopy with palm, both in 3 dimensions, in this case looking at mitochondrial DNA and its location inside of the mitochondria, in this case looking at clathrin-coated pits. So I think a lot of my success is attributable to the fact that I'm a pessimist, And I like to focus on problems because I think problems are opportunities. So I'd like to say a bit about what are the problems with super-resolution microscopy instead of extolling its virtues. First is that, as I said earlier, based on the Nyquist criterion, you need insanely high density of labels. These can cause overexpression of proteins to get to those levels, or if you use exogenous dyes, it's hard enough to get enough specificity without a bunch of background. 95% of what we look at in super resolution is fixed cells, but it's known that chemical fixatives alter the ultrastructure at the nanoscale. And so we have to put an asterisk next to almost everything that we learn by chemical fixation. What I think was a very important innovation a couple years ago was to get around the labeling density problem was Jan Ellenberg's group studied the nuclear pore, and then even though it was difficult to get perfect labeling of every structure, if you look at thousands of these stereotypical structures, you can do particle averaging techniques borrowed from cryo-electron microscopy, and then you're able to determine the positions of every one of these different proteins in the nuclear pore to less than 1 nanometer by super-resolution optics. And there was an ambiguity in the cryo-EM data as to which way the subunit was oriented inside of that, and that was addressed by super-resolution microscopy in this way, a really great example. So, of course, we heard in the introduction that Sven gave that the real promise of super-resolution, though, is the ability and the hope to look at living cells. But it's still largely a promise. Even though there's been technical demonstrations, it's been very little in terms of, I'd say, real biology learned. And the problem is that if you want to get to higher and higher resolution, you have to collect many more photons than you've ever had to do at the diffraction limit. The methods that are used today require power densities. Life evolved at 1/10 of a watt per square centimeter. We go anywhere from a gigawatt per square centimeter to a kilowatt per square centimeter with super resolution techniques. So you have to ask yourself, what are you doing to the poor cell when you're trying to look at it live? And then finally, the acquisition times of many of these methods are way slower than the rate at which dynamics is happening into the cell, so you get motion-induced artifacts or can't follow the thing you want to do. The one technique which can do much better is, because it offers much lower resolution, is SIM, which gets only twice beyond the diffraction limit, but it really offers a lot of other benefits. And, you know, it's a shame that you couldn't have 4 people in a Nobel Prize, because I think this technique is totally justified to be a part of this. So one of the real pioneers of this technology was Swedish native Mats Gustafsson, who eventually became my colleague at Janelia. So we've been working with Mats' SIM technique for a while now, and eventually pushed beyond this 100 nanometer barrier to then get to 80 nanometers, and with nonlinear SIM down to 60 nanometers, and still capable of looking at the dynamics of living cells. I think that techniques such as PALM will be good structural tools at the nanoscale, but I think this is going to be the real winner for being able to look at the dynamics of living cells beyond the diffraction limit. But it's still true that no matter what you do, no matter what method you want to use, the higher the spatial resolution you want to have, the more measurements you have to take, which takes more time and means throwing more potentially damaging light at the cell. And the moral of the story of SIM is that by backing off a bit from that limit, we can learn a lot more. So what if we back off all the way to the diffraction limit? Why would you want to do that? Well, the hallmark of life is that it's animate, and every living thing is a complex thermodynamic pocket of reduced entropy through which matter and energy is flowing continuously. So while structural imaging will always be important, a complete understanding of life requires high-resolution imaging across all 4 dimensions of space-time. And so another focus of my group is to push in that direction. And so that has led to our development based on, over the last 10 years, there's been this tremendous growth of light sheet microscopy. We've adapted this concept of non-diffracting beams, and particularly optical lattices, which was the crazy idea I was trying to work on before we dropped it for PALM, adapted that to light sheet microscopy, and now have a wonderful tool to look at high-speed dynamics inside of everywhere from single molecules to embryos over 4 orders of magnitude of space and time by this method non-invasively for very long periods of time. And so that got us back finally to super resolution, because in the same year that we published the Palm paper, Robin Hochstrasser's group published a different way of doing single molecule localization, which doesn't involve photoactivation, but just the transient binding of molecules to cells. The advantage of this method is that you can have your whole bath labeled with fluorophores that just keep coming, and so you have an infinite army of molecules and can get higher and higher density. And so by pushing in that direction with our lattice light sheet, which allows us to get high signal to noise single molecule imaging even in the background of all of these molecules in the bath, we've been able to take 3D localization microscopy up about 2 orders of magnitude in the number of localizations you can get. As you can see, like in this example, where we're looking at a whole dividing cell about 15 microns thick, and looking at about 300 million molecules, which again is about 2 orders of magnitude more than a typical image. So the final challenge, I think, going forward is how to take cell biology away from the cover slip. Most of what we know is from immortalized cells, but that's not where cells evolve. So we need to look at them inside of the whole organism. The problem is that the light rays are scrambled as you go in, and so we're now adapting techniques first developed by astronomers to make ground-based telescopes have resolution as good or better than the Hubble Space Telescope. And so in this case, you can see moving into the zebrafish brain with this adaptive optics technique, and now we turn the adaptive optics off. That's what you would see with a normal microscope, and then going back on. That's just getting back to the diffraction limit. So the ultimate goal of my group is to try to combine these technologies to be able to look deep in a multicellular context, to be able to look non-invasively and fast with methods like lattice light sheet, and then bring in super resolution techniques such as SIM and Palm to then add the high spatial resolution on top of that. At that point, I'm done and I'm out of microscopy, and I'll be back into that black phase and trying to figure out something else new to do. So, I'd just like to end with a couple things. First, there's many, many people to thank, but really, let me get this thing running right, because I want to get through those pictures. So, but the guy I have to signal out is Harold. I mean, I would have flamed out of Bell Labs in my first few years if I hadn't latched on to him as a friend and a mentor. And then there's no way I would have had the courage to pursue Palm on my own without him by my side. And so one of the bittersweet things about winning this award is not having him here by my side up on the stage. But I feel this award is very much as much his as it is mine. And then the last thing I would like to say is a lot of what you heard this morning, you know, like Suji's talk and Stefan's talk and my talk, is about taking risks. And people are always exhorted to take risks. And that's fine, but you're hearing that from guys whose risks paid off. But it's not a risk unless you fail most of the time, okay? And so what I'd really like to do is I'd like to dedicate my talk to all the unknown people out there in any walk of life who have gambled their fortunes, their careers, and their reputations to try to take a risk, but in the end failed. And so I'd just like to say that they should remember that it's the struggle itself, that it's its own reward, and the satisfaction that you knew that you gave everything you had to make the world a better place. Thank you for your time.
Please get all 3 laureates onto the stage.
Brian, thank you. Thanks.
So we thank you all for a fabulous morning. Well, I think that says it all, right? Please remember we will now have a short break.